Hello, and welcome once again to Propel video series, where we interview innovative scientists about their work. Today, we are thrilled to have the founders of Promakhos Therapeutics, Dr. Katerina Chatzi and Jasper Neggers. Last year, Propel made a call for proposals, and out of 9 well-deserved projects, we chose Promakhos. Dr. Chatzi is a distinguished scientist entrepreneur with expertise in immunology, protein secretion, biochemistry, bacteriology, cell biology, and business development. She has performed biomedical research for over 15 years across 4 different countries, uncovering fundamental principles underlying bacterial secretion, protein folding, antibiotic resistance, and cell cycle regulation. She earned her PhD in Greece and has worked in Sweden, Belgium, and in the United States at Harvard University. Dr. Neggers hails from the Netherlands and completed his PhD at the Rega Institute for Medical Research at KU Leuven and Belgium. Dr. Neggers is an entrepreneurial, pioneering, and innovative scientist with a background in microbiology, immunology, cell biology, target identification and validation, drug discovery, and functional genomics. He was among the first scientists to apply CRISPR-Cas to genetically engineer human cells. Dr. Neggers arrived in the USA as a postdoctoral fellow at the Dana-Farber Cancer Institute and at the Broad Institute of MIT and Harvard to spearhead a team of researchers to identify and validate new therapeutic targets for cancer. He quickly distinguished himself and was awarded a Damon Runyon Cancer Research Foundation Fellowship. His work has nominated several new high-priority therapeutic targets for cancer and has led to the initiation of new drug discovery campaigns. As a rising scientific leader, Dr. Neggers was on his way to become an assistant professor after receiving the National Cancer Institute Pathway to Independence Award for outstanding early-stage postdoctoral researchers. However, ultimately, Dr. Neggers, wanting his research to translate into patient impact, felt that an academic career would not provide the right environment to pursue his vision. So incredibly, he handed back his $1 million grant money and joined Katerina to co-found Promakhos Therapeutics. Thank you both for being here today.
Thank you.
Thank you for having us.
I'm very happy to be here.
My name is Angela Clark with Propel, and as Annabelle said, we're so happy to have you both finally for a sit-down chat about your wonderful work with Promakhos and to introduce you to our viewers. And before we dive into the substance of all the work you're doing, I have to say you both had very distinguished careers in Europe before you ended up here in the United States in postdoctoral roles. What inspired you to establish Promakhos Therapeutics, which is a biotech startup, and what was that process like?
Maybe I can start. So, in academia, you're usually studying a very narrow subject, very deeply. But when you see something through your academic career, which you think that it could be translated in humans and make human lives better, this is something that really excited me. And it felt the right thing to pursue that and start Promakhos Therapeutics. It's just very exciting to dedicate your hours knowing that you're developing something that may drastically improve patient lives. It's just very fulfilling.
Yeah, I can add a little bit more to that because in an academic setting, especially for younger career researchers, it is sort of promoted that you continue in a very narrow, specific way of looking at a problem. And once you have published the paper, you're not encouraged enough to actually take this to the next step and develop it in something new. And typically this involves getting new skills or starting collaborating with other people. And it is in a certain way, it is promoted that you do more with it, but not in a way that always benefits the patients. And you are sort of, I'm sure you have seen this, that sometimes in academia, something gets published that is interesting or very exciting. And then people discuss it in a meeting and say, oh, very interesting, we should do something with that. And then a year later, nobody did anything with that. And they all start doing something else. And this is maybe slightly different in Europe than in the US. In the US, it is promoted more to take these things to the next step. At least I have this feeling in the Boston environment. And also there is a richer environment that is able to support with money or funding or from investors or from patients to actually help people develop something. This is harder to do in Europe than it was here, but it still felt that within an academic setting, even here, it's not always possible, and it's making a company helps maybe to set the mission clear that you want to make something for patients. It sets you up also in a more difficult position because you develop a particular product, and if after 8 years in the clinical trials it might not work out, that is sort of the end of the company and you have to start something new. Whereas in an academic career, you try to have your ropes open and go around and collaborate with others. And if something doesn't work out exactly, you start doing something else and you sort of keep your options open in that sense. So in that sense, it's a risk aversity that drives academia in a certain way that is not in companies, but it does have more risk. So that is the downside of making the company. Sorry for this detailed answer, but that's my personal feeling.
No, I'm so glad you mentioned that, because that's one of the common themes we see in IBD research that have frustrated us as patients and loved ones as we've watched it, that you see all these studies and then that's it, and we're so thankful you're on the translational end of it and trying to bring new exciting developments to us through your research. We're excited to be helping fund your study. I want to get this right, so I'm going to read it, which is called Validating the Role of Mucosal Innate Immune Deficiency in Crohn's Disease. Now, what do we mean by innate immune deficiency? And what led you to believe that it would better help us understand why Crohn's disease happens?
So the innate immune deficiency that we're specifically mentioning is in the gut. So the gut is an ecosystem where you have the outside world where our food and actually the bacteria of our gut live, and they interact with our body in the first layer of the tissue, which is called the mucosal tissue, and the epithelial layer. And in this interface, these microbes in our body are in a constant flux of trying to balance the needs of both. So the microbes live in harmony with our body, but we cannot let the microbes overgrow, because then they take over and they become dangerous or infectious. And there is this finding, especially in Crohn's patients, it was originally discovered in the 70s, that there is a deficiency in this immune response against microbes that keeps this balance intact in neutrophils, which is a specific type of cells that are in patients. And this was sort of developed into an idea that maybe there's a problem with this innate immune line. But then it went to the background because the tools that we have to study these innate immune cells in this first layer were not developed as much as they are now. And then in the early 2000s, when genetics came back, it became clear that many of the risk factors, the genetic mutations that are associated with Crohn's disease, are related to innate immune genes or functions. So there are a lot of genes like NOT2, like ATG69, like IL-10, TLR4, that all have to do with initial immune responses against microbes. So the first layer of our immune defense to maintain these microbes from invading our gut. And these deficiencies were discovered genetically, and then there was a resurgence a little bit in studying this innate immune deficiency. There were some great papers in the early 2000s, but there weren't any drugs that were able to really address this problem, and it was more observational studies in patients. And at the same time, all the other elements of the immunology came to a full maturity, I would say, which was on the adaptive immune system, on the T cells, which are more part of our general body immune response and not always unique to the gut. And a lot of therapies were being developed in that field that actually started working, and then this started going back to the background again. But now, after this 10, 20 years since these drugs have been developed, it has become clear that these have reached the ceiling of efficacy. They cannot become better. So many more of these older ways of, the new way from the 2000s of targeting the adaptive immune system is not working out. And now it's clear that something else is causing the disease, or there's an underlying problem that is upstream of these adaptive immune cells. And we are based on this position that it's the innate immune layer that is defective, and that is what we actually have to restore. So I don't know if that answers the question, but that's sort of the background of the story.
Are you finding that, or do you think that all Crohn's patients, are you seeing that they all have it, this innate immune deficiency, do you believe?
I think definitely the majority. So it could be as much as all. You could say that different genes are in different patients, but most of these genes relate to innate immune deficiency. And then, of course, there's a non-genetic component. So genetics maybe explains 20% of the patients. But then there comes the microbiome. And the microbiome itself releases molecules that activate or train our innate immune system to actually be in balance with these microbes. However, we discovered through a collaboration with the Brigham and Women's Hospital at the Harvard Medical School that a certain set of these metabolites that are released by the gut microbes that actually train our innate immune system are not present in 80% of patients, or present at way lower levels than in healthy volunteers. So if you combine these two elements, you could come to almost 100%. So you have 20% genetic reasons why innate immunity is not effective in Crohn's patients, and then this 80% could be because the metabolites in the bacteria are not being released and our innate immune system is not activated and trained properly, and this is causing this innate immune deficiency in this class of patients. So there is an overarching theme that it could be 100% of all Crohn's patients, but it could be in different ways. Certain genetic elements could trigger the innate immune deficiency in certain patients, certain reductions in the metabolites from the bacteria in other patients. So it's not there's one underlying cause for the innate immune deficiency, but the innate immune deficiency seems to be consistent across these patients in this particular studies. So there is a reason to think that it could be up to all of them.
And what happens? So walk us through when there is this innate immune deficiency. What do we see happening in the body?
So what happens in this case is that in the gut, the bacteria start overgrowing and infiltrating the tissues in our body, and this is obviously not good. So it becomes a bit like an inflammatory, infectious phenotype where the bacteria are present in our gut. But then the backup systems of our immune system start try and compensate for that. So if this initial layer is failing and there's an invasion of the microbes, you have other mechanisms that can start being activated, which include the adaptive immune system, but also components of the innate immune system that come from the bone marrow and from the bloodstream. So they are recruited, like the fire department, they go in the side and try to maintain and contain the problem. So they get activated together with these adaptive immune cells to try and prevent this bacterial overgrowth. But they are not sufficient enough because this initial layer is defective, and without it, they cannot sort of remove the bacteria as efficiently as in healthy patients. And even if they would be able to remove it, maybe then after some time, it comes back because this innate immune deficiency is still there. So maybe you get rid of the infiltration and they are gone. But as 6 months later, there's a new wound or a new infection at a different site. The innate immune system that is present locally is not working, so you get a re-trigger, you get another inflammation, you trigger again the secondary response, and you get a flare-up. So in that case, it worked initially, but it comes back later, and you get a flare-up. In the other case, it could be so dramatic that these secondary systems are not sufficient, and it develops into a chronic worsening phenotype that just doesn't resolve. So then you get like more severe or chronic disease where it just becomes worse and worse over time, and we're not able to actually get rid of the initial bacterial stimulus. But again, this is a particular theory that our company is based on. There's a lot of evidence since the 70s, and I don't want to say that we are 100% sure that this is the cause of the disease, but it's one particular theory that is gaining ground that we have based the company on. So that's just one thing I want to make sure that I'm saying.
Well, having said that, the reality is that Crohn's is not a genetic disease. And the chance is that we will only know what's the real cause of the disease once we have either developed a cure or something that works really well, a drug that works really well. And then we'll say, oh yeah, if we activate or block this pathway, then we have such a good outcome that probably that's the cause or very close to the cause of the disease.
On that note, this is going to be tricky, but can you summarize the study that Propel Cure is helping to fund and your progress to this point. Now, you started out studying stool samples from Crohn's patients and have made some exciting findings along the way. So please take us through that, and then where the study goes from there, if you would.
Yeah, so our initial finding was that there's certain metabolites that are present in the gut microbes and that are released towards our body and our gut were not present in Crohn's patients, and a reduction of maybe 4-fold less, and in some patients almost non-existent. But this was in a study with 25 samples, and that is when we made our proposal to Propel Cure to basically expand on this amount of samples that we have by adding additional samples from additional Crohn's patients in different types of stages of disease, and validate that these metabolites that activate the innate immune system are indeed reduced in a larger cohort of patient samples from fecal samples from patients. Now, we have done that in the last 9 months, and we are happy to announce that our main findings are maintained. There is a strong reduction in up to 80% of the Crohn's patient fecal samples in these bacterial metabolites that can be 4 to 5-fold less on average in patients with active disease, and some patients have very low levels that are almost non-present. But these are individual patients. So that's what we have done. It confirmed our initial findings. And now we are taking this one step further, and we want to use this patient sample that have these low levels of the metabolites in a mouse model to develop a new mouse model of Crohn's disease where we use these fecal samples with these low levels to mimic the clinical environment in patients where these metabolites are present at low levels, so that the innate immune system is not properly activated in these mice, and then induce bowel inflammation in the colon and the ileum, and then see if this is worse when you have lower levels of these metabolites compared to higher levels. That would validate the concept that low levels of these metabolites make inflammation worse in the gut, could be the trigger of Crohn's disease. And then on top of that, we want to test one of our developed therapeutic agents in this mouse model to see if we restore this signal. So this therapeutic agent mimics this bacterial metabolites, and we add it to the mice to restore the levels of these metabolites to normal levels so that we reactivate the innate immune system, if that could treat the bowel inflammation in these mice. So that's what is next. That's coming up for the next 9 months.
As a small addition to that, so we're using germ-free mice for this study. That's important because these mice have never seen any microbes in their lives. So they are being transplanted with the microbes from the patients. So we're also reflecting this component than the ligands that just described. We're also reflecting the microbes. So we think that this model will mimic better what happens in the patient, or at least some aspects of the disease in a better way.
Yeah, that's exciting news indeed. And now we understand, since the study has started, you have attracted some other collaborators, other institutions that you're working with. Can you tell us more about that and what part they're playing?
Yeah, so to evaluate our therapeutic agents further in a patient-relevant context, we have started a collaboration with the University of California, San Diego Humanoid Center of Research Excellence. This is led by Dr. Pradipta Ghosh and Courtney Trindle. I think you have a video with them, so anyone that wants to know about them, you can look at their video. It's from 2 years ago, I think, and it's on the Propel Cure website. They have established a organoid biobank, which is they took biopsies of Crohn's patients from the colon and cultured these cells from the Crohn's patients in a lab system in a way that really reflects the Crohn's patient biology without losing aspects of this disease within the lab setting. And this allows us to test our therapeutic agents in a humanized, simple laboratory test that reflects the Crohn's patients with the actual tissue from the Crohn's patients. And we are using our molecules to show that we can induce bacterial maintenance and clearance. So the idea is that in this particular model, we can use our molecules to see that we rechange the balance against certain microbes, validating that this activation of this particular innate immune pathway is important in regulating the microbiome, which is definitely something that we think in patients is also important for our therapy. So that's one collaboration. The second collaboration that we have started is in Belgium at the KU Leuven, which is with the Center of Dr. Professor Severin Vermeer and Brian de Stokt, and we are expanding further on our initial study with the fecal samples from patients where we have identified that these metabolites are reduced in Crohn's patient samples here locally from the Brigham and Women's Hospital. And now we want to sort of repeat this study in Belgium with a large collection of stool samples from patients and confirm that in a different area in Europe, this phenotype is also present. And on top of that, they have additional genetic information that can further inform us if certain subpopulations of patients have even lower levels of these metabolites compared to others. So that's an extension of what we have already done in a different area that will hopefully validate the concept even stronger.
I think that speaks so well to the research you've done and the results that you're seeing and just the excitement that this is generating, that you're able to attract interest in other institutions that want to collaborate you, and we're just very, very thankful for that. What kind of time frame are we talking about? So you're moving into phase 2 of the study. For the conclusion of this study to human clinical trials, for example. I know people are always wondering about that.
So for our particular agent, we probably need 2 to 3 years before we can actually test it in humans. And then we first have to validate that it's safe and follow the FDA regulations before we can test it in patients, which might take an additional year on top of that. And then we can test this therapeutic approach in Crohn's patients, in a small set of patients initially. And then once this shows promise, we can go to a larger set of patients and then sort of get approved from the FDA after that. In the setting of how we are envisioning that the current studies can help us with this clinical phase is that many therapies now for Crohn's disease in particular, they work in some cases, but not in all cases, and it's very hard to predict when they will work and when they will not work. And there's been a lot of effort in trying to find biomarkers that can predict whether a certain therapeutic agent will or will not work in a certain patient. However, it has turned out to be really difficult to find promising biomarkers to predict therapeutic response for these current agents. And partially that is maybe because they are not exactly addressing the cause of the disease and the biomarkers are just not established to validate that, but it is also that it's difficult to really do this work. Now, with these patient samples from the stool samples in particular, we could, if our findings validate across different sites, this level of the metabolites that is present in the stool of patients can be a great biomarker to predict which patient will respond best to our therapeutic agent, because you would expect the patient that have the lowest levels of this metabolites will benefit the most from the drug, because then it reactivates this the most. So we can maybe select the patient that will benefit very well initially, and then go later to expand to additional patients and see if it works also there. But it can actually help us really select patients and say, okay, this might be a therapy for you, or this is not as great as for this patient. Now, that said, we still think it could address a large population of patients, maybe up to 80%, but this will need further refinement during our next few years as we expand our studies with these patient samples and also start doing work in the clinic in patients. So that's one aspect that I definitely feel that we are at least taking into consideration.
So coming back to your question about when it can be tested in humans, we are quite close to finalizing the molecule that we think it is appropriate to be tested in the clinic in humans. And we think we can achieve that by the end of this year. And then it will take another 9 months to a year to do this extensive safety studies that the FDA requires in animals to show that this is actually safe to be tested in humans. And then we will be ready to start phase 1, which is in healthy volunteers, hopefully in early 2027.
That's wonderful.
Sorry. So this molecule that you're talking about, this treatment, do you envision that it will be something a long-term that somebody with this deficiency will have to take, for the rest of their lives, or will it be something that will be able to just sort of fix things and then that will be that?
So initially, because we have to start with the most conservative scenario, we will test it in the context of a patient takes it for 12 weeks and then for a full year and see how the disease progresses. And this will really inform us if that would be a treatment that you have to take for the rest of your life, or maybe not. Now, because this pathway that we're targeting directly affects the microbiome, or at least it can affect the microbiome, and we have data about that, there is a chance that after you take this treatment for sufficient time, you see some recovery even without taking the drug. But that's something that remains to be tested.
And I just want to reiterate, we're talking about a non-immunosuppressive oral therapy that you're hoping comes. Okay, wonderful. Now, Dr. Neggers, I wanted to ask you, because of your background in cancer research, how has that helped your approach in solving the unknowns behind Crohn's disease, or have you been able to bring anything to that from your background in that? And are there any parallels between the two that you can share?
So what I did like from cancer is that cancer tends to have very genetic elements that are quite specific to a particular cancer, but that helps you stay focused and kind of keep the approach a little bit aligned with a particular way of going forward. And that is something that in Crohn's disease has helped me specifically, because we work on a very particular target, and trying to understand the biology of that target within cells individually at mechanistic molecular level does help you develop drugs. Whereas the downside of that is that in cancer now, they have expanded to immunology, and that definitely opened up a new way of thinking in cancer, and that was needed in cancer. But in Crohn's disease, obviously, that was already the case. It was always being approached as a systemic disease. It has different components with the microbiome, different components of the immune system, and we have to see it as a whole. I was fortunate enough that during my time in cancer, that was sort of how people started changing their view about cancer, and those learnings of how we reimagined that and how to use the immune system within cancer has helped me to keep working in Crohn's disease and keep best of both worlds. So the systemic immunology component, but also sort of a targeted approach that you really focus on a particular element and try to get into all the details of that. Those two combined, I have been applying that within the Crohn's field, so far with success in the company, I feel. And that is definitely something that I sometimes felt that if you're not as familiar with focusing on particular targets in drug discovery, you can get lost in showing, oh, this immune cell does that, this immune cell does this, but you're sort of looking at symptoms and different ways of immune cells doing things, but you don't try to make a full story, a single story that tries to just explain from beginning to end, like it is the progression and this is where it started and this is how it developed. So that is the benefit, I think, that I had in one way.
Have you run into any obstacles or surprises during this particular study? And does anything come to mind?
As a company, but this is something that I think everyone has felt in Crohn's disease research. I originally had a background in pancreatic cancer and colorectal cancer. And pancreatic cancer, many people know pancreatic cancer. Everyone knows it's a very deadly disease. There's a survival rate of 90% for 5 years for certain classes of patients, a mortality rate of 90%, not a survival rate, sorry. So that is really drastic, and everybody in the world knows that. I go on a plane, and if I say I work at the Dana-Farber Institute, then they're immediately, okay. And then I say pancreatic cancer, and they're immediately all over you, and they're like, okay, this is a very deadly disease, this is very important. In Crohn's disease, many people have heard of it, but this urgency across the world that we need to do more, or there needs to be more funding, or there needs to be more initiatives to support it, is less there. And I feel that this is a mission for us and maybe for everyone, but I feel that we need to make the world a little bit more aware of how serious this disease can be to certain people and how important it is, and that there needs to be more funding to try and develop approaches for these diseases. And I think as a company, there's also one other thing that I have noticed is that once you leave the academic institute and you become sort of your independent company, it becomes initially harder to get validation of what you're doing, but then when you start collaborating with academics, it starts coming back again. And there is this fear that once you go into a company or industry, that for certain academics, that you're outside and you're a weird person and you have to be careful when you work with them. But we're still the same people here when I was in academia. So that felt a little bit weird. But that was maybe in the beginning, and as time goes by, it becomes less of a problem. But that was initially definitely something that I experienced.
Yeah.
It's definitely reflected in the funding also from the NIH or the Department of Defense that there isn't as much funding for Crohn's disease as it is for other, even some very rare diseases, which reflects partially the awareness of the people about the seriousness of the disease that has, of course, reached the politicians that actually make these decisions.
Yes, we share your frustration too about the disease, and that's part of Propel Cure's mission, of course, is to really bring home to people how serious it is. And we're continuing to work on that. Now, so you're focusing on the innate immune deficiency in Crohn's. What part do you believe environmental triggers may play in this process?
So we think that the bacteria, the gut microbes, are sort of the underlying inflammatory trigger that happens in Crohn's disease. This is sort of well established that the microbiome is obviously a major factor in disease development. Now, I think that this explanation of the innate immune deficiency is elegant in a way that it allows you to not say that it's a particular microbe. So it's not an infection of one particular microbe, like it's C. difficile, or is this bacteria, that Mycobacterium paratuberculosis or something like that. It's not. It allows you to say, okay, this innate immune system is there to respond to all microbes in a way that it remains in a balance, but it's not selective to particular microbes. So if this innate immunity is deficient, it allows different types of microbes to maybe become pathogenic or inflammatory. And this is, in different people, can lead to slightly different disease outcomes or disease development, and explains part of the heterogeneity that we see in Crohn's disease that has not been particularly explainable if you approach it from it should be a particular antigen or a particular bacteria. It could be that it's a wide spectrum of different bacteria that happen to be there in certain patients with one type of bacteria, but in another patient it's a different type of bacteria. But because this innate immune deficiency is there, both of these bacteria infiltrate the gut and become inflammatory or trigger an antigenic response. But it is slightly different in these patients because the actual bacteria or the bacteria, it could be more than one in a particular patient, is different or unique to that patient. So the details of the exact response that happen from the T cells and the innate immune cells that are recruited can be different, and the disease development is different. But it can explain everything at the same time. It just is that maybe if you restore this innate immune defect, all of these microbes will go back into the general area of the gut and not be pathogenic, and you restore the balance without having to fight one particular bacteria. So that's part of the beauty, I think, of this story.
So that it won't matter what's actually, and it could be different things in different individuals. And with what you're looking at, there's the potential there to address all those variations, it sounds like.
Yep.
Great. So circling back to, it's so interesting what you're talking about, the companies. Can you, being a startup yourself, what role do you, and we've had questions from supporters that don't quite understand the relationship between companies and academia, and what role do you see the startups like yourself playing in finding answers to Crohn's disease? Because it's a really exciting area, and a lot of people don't know much about that. They're used to just the academic institutions.
So usually in academia, right, there's done research to figure out the pathophysiology of the disease, and specifically for Crohn's, there is a lot of research that has been done out there, and some different theories about what could cause the disease and how you're going to reverse it. So I think that this field, the Crohn's field, has reached to a maturity that we should be testing different ideas in the clinic. And we should be developing different drugs that address different hypotheses about what could be causing this disease. Right now, we have focused that the drugs that have been approved and the drugs that are mostly the pipeline to be tested or are already being tested are around the idea that we have to suppress certain proteins or aspects of the immune system, usually around T cells, suppress T cells one way or another, or some aspects of the innate immune system. But there are other theories out there. This approach has really been exhausted. Like all our resources have been put towards there. But there are, other very interesting theories like the one that we are testing, that maybe Crohn's is an innate immune deficiency, and that's a completely different drug development approach, right? Or another thing that's very interesting is the microbiome, right? That can we change the microbiome, and then we may be able to cure the disease. So we are talking about microbiome transplantation approach. Or can we fix the gut barrier, right? Because that's aspect of physiology. Let's fix it and maybe we'll cure the disease. But all these later ones are not being tested in the clinic, so we will not figure out, actually, as I said in the beginning, if we don't have some progress with the disease in the patients, we will not figure out what's the real cause of the disease. Usually we were thinking that we are going to study it in academia, right? And that's like study the disease in animal models and from patient samples. And that's what will lead us, if we study for many, many years, this will lead us to know what's the root cause of the disease. But there is the chance that we will learn what's the root cause of the disease only if we test different approaches in patients. And then we see, okay, that's the one that's way more successful. And then let's put all our resources towards making drugs towards this approach. And then I think we may have a cure or a very, very good treatment. So I think that's back to your question, what's the role of the companies? I think that the companies should test different ideas that come out of academia and really put the tremendous amount of energy that is needed to actually take this drug development approach forward.
Yeah, I agree with Katerina. I think the role of the startups is to focus on a particular idea and try to really develop this into a drug and see if you can make it work. Because to some extent, only if something works, you have proven the fact that this was actually the cause. Because unless you sort of prove that if you fix this thing, and you can always speculate it could be the cause, but unless you show that it actually is treatable, you can never really validate the hypothesis. So there's this issue that you can research something for a very long time and describe everything you see in very excruciating detail, but at some point, someone has to test it in a therapeutic intervention to prove that it was actually all along wrong or right. And that is the reason why you have companies. It has its own nuances that are maybe difficult or specific, because you do have to balance between an academic setting and the VCs and the investment community, where profits and returns on money become important. So you have to balance these two things. But startups are in a position where they can identify with a unique approach or unique vision that more so than a larger pharma company can. They are more restricted in changing their image, or they have a certain image that is very hard to change. It is very slow to change. So they have this problem that they have to take it step by step. They have multiple layers that have to be approved. They're typically also very tightly watched by politicians. Like if they do something wrong, they get immediately like, oh, what's going on here? We have to get to the Senate. So these companies they are in a more difficult situation where they cannot take up new ideas that are a little bit risky as easy as startups in that setting.
And the other reason why it's difficult to bring new ideas, because we have, as humanity, we have ideas, but it's difficult to get them to be tested, because it requires a lot of capital. And the other thing is that when you have new ideas, many of the new ideas are going to fail, right? So justifiable to some extent, there is a risk aversity from the funding agencies, from the stakeholders, to avoid these very new ideas and instead go and create and generate something that is closer to something that has already worked. So that's why I said that it's a tremendous amount of energy to persuade all the stakeholders that a new idea is actually worth testing.
And there is definitely a role for patients and patient foundations and clinicians in that. And that's actually why I'm very happy that we were able to engage with Propel Cure. Because within a company, if you just focus internally on developing something and test it in the clinic, you will get a lot of criticism from larger funding agencies or larger pharma, like it's risky, it's a new idea. So you need this kind of energy where you can connect with patients that sort of tell you, look, what you're doing, we know there's a risk, but it's definitely something that addresses a real need in patients. And we are really supporting you, and we have this momentum behind you, and you will maybe able to help actual patients live better lives. So that's definitely a motivating factor from the patients and from your foundation as a whole. And then on the other side is the clinicians. We have been fortunate enough to work with different clinicians at the Brigham and Women's Hospital, the KU Leuven, now in University of California, some people that have worked there. And it is very rewarding to see that clinicians are willing to take this collaboration with us to work on this idea, to at least help us move forward and try different elements that can help us develop these drugs further, and trying to assist with their unique tools or unique materials to at least help us move forward, and also drive the masses that patients are rooting for this kind of drugs and they need new drugs. So you have both the clinicians and the patients that keep this energy going, keep this motivation and this inspiration to make an impact going, because without it, it can become a little bit lonely. It's something I alluded for in the beginning, that within a company, you're now not in the larger institutes anymore, so you have a little bit of a barrier to collaborate. People are a little bit more hesitant. And the same is with like larger funding institutes or larger pharma. You're a small team, you haven't established a reputation, you're not Harvard, so they're not as, it's a new company, so they're a little hesitant. We don't know if they have proven themselves. They exist for this amount of years, so they have to take it step by step. Whereas in these larger institutes, you have the benefit that it's easier to work with them from the get-go. And here you have to take it step by step and take them through your idea one step at a time. And then if you are good enough, they will work with you, but it requires this additional patience to take it step by step and keep inspired along the way. So that's definitely something I wanted to tell you.
Something else that I have realized about patient advocacy, it's extremely, for chronic diseases, patient advocacy is extremely important. Your role is very important and what you're doing. And it goes, other than all the things that Jasper mentioned, right? Even at the level of knowing if is a new treatment needed, and if this new drug that you're developing works in the clinic as you imagine, will the payers, the insurance companies, reimburse it? The insurance companies need to know if the patients really need it, or if it's just, some people that are just developing new drugs. And that's also where the patient advocacy groups are very helpful, because if you know that the insurance companies are going to pay, then the pharmaceutical companies, they want to develop that. And then eventually funding goes towards startups to actually develop this new approaches.
Thank you for enlightening us on all these dynamics. It's so interesting, so important, and really needs to get out there. And since you touched on it, you touched on the difficulty of getting funds and investors and bigger funding entities not wanting to take risks so much, and to the extent that Propel Cure donors are funding some of your actual research. Can you just tell us how that has helped you all in this research project?
I think it has been very inspiring for us. Actually, when we heard that we had won the award, we were really happy. I can tell you that. it was also the first time we actually engaged with you as a foundation, and we realized that patients are really looking for people that are willing to stand up and take risks and develop something new, and that was really inspiring. It has helped us communicate to others, like, okay, look, we have talked to patients, you have some recognition in the world, and people definitely realize, Propel Cure is interested, that definitely helps validate that we are doing something that is worth trying. And that is definitely something that has helped. This wasn't as clear in the beginning when we just received the award, but it has become clear in the last 3-4 months that it definitely has a beneficial outcome in that sense as well. So that has been really helpful.
And because, at the end of the day, the funding comes down to having data. This funding that we received from you, it all went to generating data. And this is what we are going to use to get more funding and continue developing this drug.
Yep.
And I know that was a new thing for me when I first became involved in this, people not knowing that you have to have data to attract funding from the really huge funders. And that's why I think the donors need to know what they're doing now is so critical in these early stages, providing this really vital funding. And we're so happy to be able to do it with you all and appreciate so much what you're doing. Annabelle, do you have any more questions?
No, but I'm really pleased with this because I think you've done a great job answering it. I mean, there were a few questions I had, and then you just answered it. So I don't really need to ask anymore. I just want to reiterate what Angela said, that we are thrilled to be helping you guys out. As our supporters know, I mean, as all of our supporters, we feel very strongly, and we need hope. We're frustrated because we feel like it's been sort of this stagnant time where there just hasn't been a lot of new stuff coming out to help us. And I think that what you're doing it's going to change things, and it's going to make a totally different landscape there for us. So we're really very excited, very, very excited. And every time I talk to you, I get excited. So that's good.
And we will be seeing more updates from you all. I look forward to that. Of course, we bring them to our supporters in our newsletter, in our social media, in our emails. So we want to thank you, Dr. Chatzi and Dr. Neggers, again for everything you're doing, for giving us hope, the whole IBD community hope, for inspiring other researchers and finding new answers. And I think you will get there. We'll certainly be watching with excitement. And thank you for being with us today.
We're very grateful you're supporting us. Thank you very much.
Yes, thank you for inviting us also for this interview. And we're very grateful to all the support you have given us so far.